Hello and welcome to the November edition of the ASC e-Journal presentation. My name is Dr. Rezai. I am the current chair of the e-Journal committee and I will be moderating today's session. It is my pleasure to briefly introduce Dr. Walia, a member of this committee who is presenting the title article. Dr. Walia is an assistant professor of pathology at the University of Southern California with clinical and research interests focused on, obviously, cytopathology, but also GYN and breast pathology. We are also very grateful to the first author of the article to be presented, who accepted our invitation to participate in the discussion, and Dr. Walia will introduce our guest shortly. Lastly, and in the spirit of having an interactive presentation, we encourage audience participation, but please send your questions and comments through the question box, and we will address them in the last 10 minutes or so at the end of the presentation. With that, I will turn over the presentation to Dr. Walia. The floor is yours.
Thanks, Dr. Rezai. I am very pleased to be presenting this very timely article that came out that's titled Molecular Testing Opportunities on Cytology Effusion Specimens, Pre-Analytic Effects of Various Body Fluids Cytology Preparation Methods on RNA Extraction Quality and Targeting Sequencing. This came out of multiple institutions, MD Anderson, UT Houston, and Yale being some of them. And like Dr. Rezai mentioned, I'm very pleased that the first author, Dr. Sura, could join us today for this presentation. Dr. Sura is a board-certified physician in anatomic and clinical pathology, cytopathology, and molecular genetic pathology. She's currently working as an assistant professor in the Department of Pathology and Lab Medicine at the University of Texas MD Anderson Cancer Center. She has completed her residency training in anatomic and clinical pathology at Louisiana State University Health Sciences Center, New Orleans, followed by fellowship training in cytopathology and molecular genetic pathology at Houston Methodist in the Texas Medical Center of Houston. Her research interests include pre-analytics of molecular cytopathology testing, predictive biomarkers, and personalized medicine. Welcome, Dr. Sura.
Hi, thank you. Happy to be here.
So this article, like I said, is very timely. The objectives of this article is to basically compare various pre-analytical sample methodologies that can affect RNA sequencing. So most labs across the nation are using different cytopathology preparations for preparing their effusion fluids. Formalin-fixed paraffin-embedded sections are most commonly used for molecular testing. However, there are other cytological preparations like Carnoy solution, ethanol, air-dried smears, as well as liquid-based preparations, including thin prep and SurePack, that can be utilized for this testing. For DNA sequencing, it has been shown that smears that are fixed in ethanol do provide good quality material for DNA sequencing. However, similar studies have not been performed for RNA sequencing analysis, and the objective of this paper is to provide the data and analysis for this. To do this, the authors measured RNA quality. They also measured multi-gene expression signatures and activating mutations in each of the 6 preparations that are mentioned here. So just to give you a brief introduction about this, RNA sequencing analysis is emerging as a clinical research and diagnostic approach for cytologic samples. And like I mentioned, formalin-fixed paraffin-embedded tissue is the go-to for most labs for molecular testing because of its familiarity and also availability and easy storage. However, we do also have to remember that formalin fixation can lead to rapid nucleic acid degradation and can also cause chemical modification to nucleic acid. This can make it difficult for nucleic acids to hybridize and amplify and make molecular testing more difficult or less effective in formalin fixed tissues. So we'll just go over a few definitions that are being used in this article to get the audience familiar with what is being said. First is RNA sequencing analysis. It basically is a methodology that measures gene expression, and this can be done for the whole transcriptome, or it can be done for selected transcriptomes. And we can use archival pathology samples for doing this testing. This testing involves sequencing billions of bases of RNA across across tens of hundreds of paired or unpaired short reads. So the data that is thus obtained is bioinformatically realigned, and it is assembled to measure expression of thousands of RNA transcripts. So what it does is it can provide us quantification of known or predefined RNA species, or it can also help detect and quantify rare and novel RNA transcript variants within a sample. In this study, the authors did use two assays, which is the first one of which is the SET-ER-PR assay. This assay is used in patients with metastatic hormone receptor positive, HER2 negative breast cancer, and this provides information about the benefits of endocrine therapy in these patients. So for this assay, the authors have already done prior studies showing its effectiveness for hormonal treatment. They use estrogen receptor and progesterone receptors transcripts and measure 18 informative transcripts for these receptors. In addition, 10 reference transcripts are also measured, which are based on minimal variability and high reproducibility in each sample. And in addition, the ligand binding domain of the estrogen receptor is also sequenced using RNA-seq. It has been shown when the SET-ER-PR value is elevated based on a calculation, it can lead to prolonged sensitivity to endocrine therapy in patients with metastatic disease, especially when there is an absence of mutated ESR1 transcript. The second assay that the authors have used is a SET4 assay, which is used in stage 4, metastatic breast cancer, hence the name SET4. And it includes, in addition to measuring the estrogen receptor and progesterone receptor transcripts, like I previously described in the last slide, it also measures other targets that can be used, like the PIK3CA mutations. So they look for 9 gene transcriptional signatures in this gene, and also look at point mutations and expression levels of other genes, as are mentioned in this slide. Coming back to the paper, for the purposes of the gold standard to see what these samples should be compared against, fresh, frozen, high-quality RNA samples were utilized that were stored in RNA preservative called RNA later. The 6 cytological preparations that were used to see what kind of RNA quality, what kind of transcripts, and whether mutations would be detected were the fresh frozen paraffin embedded cell block, cytospin samples that were fixed in carnoise solution, cytospins fixed in 95% ethanol, cytospins that were air dried and Diffwick stained, and then liquid based preparations like thin prep and SurePack. So each of these 6 samples, the quality and the quantity of RNA extraction was calculated. In addition, also the transcripts that were available after sequencing, and also the mutations were seen. So the objective again of this paper is to determine the optimal pre-analytic cytological preparation condition and extraction methods for successful RNA sequencing based molecular diagnostic testing. So to achieve this, the authors chose 13 malignant effusion samples that contained metastatic breast carcinoma. These samples were submitted fresh to the lab for routine processing. They were stored at 20 degrees centigrade for an average of 24 hours before processing. So here I'd like to ask you a question, Dr. Sura. How did you choose your samples for this paper?
So we chose effusion specimens that contained metastatic hormone receptor positive breast cancer only. As for tumor cellularity, we did not determine a strict cutoff, but in general, the tumor cellularity on the slides was at least 10 to 20%. Also, it was imperative that enough fluid was collected in order to be able to create multiple slides of each preparation type.
Okay. Awesome. So, I did a Twitter chat and poll yesterday, and this tumor cellularity did come up quite a lot. So, the question that I asked was that what pre-analytical, what are the different pre-analytical things that the pathologists are using before they send out their samples for molecular testing, and almost 100% said what they were looking for is tumor cellularity. Very few or almost nobody said that they were looking at what kind of fixative that the specimen was being stored in. So that was something that was not on top of mind for everybody, but it was more tumor cellularity. So the authors do provide a very detailed material and methods of how they perform this testing, and it was very nice for me to read to see how this lab is doing their tests, to compare to what other labs our labs do. Like Dr. Sura mentioned, they had effusion samples with enough quantity. So the quantity ranged from 600 to 1,000 ml. That was filtered through gauge to remove fibrin. And then cell pellets were prepared using centrifugation. The cell pellet, a quarter of the volume, was then given over for cell block preparation, and then the remainder were used for other cytological preparations. For a formalin-fixed paraffin-embedded cell block, these were fixed in a mixture of 10 ml of 10% buffered formalin and 95% ethanol between 8 to 48 hours, and then processed the cell block. They used 10 micron thick sections, which were mounted on glass sides to be used for molecular testing. For the remaining cytospin and liquid-based preparations, the remaining cell pellet was resuspended in RPMI. 18 cytospin slides were made, 6 each for Cornoy solution that was stained with PAP, 6 for 95% ethanol, again stained with PAP. 6 slides were air dried, then fixed in methanol and stained with the Diffquick stain. For liquid-based preparations like thin prep and sure pack, 3 drops were put into the proprietary container, and slides were made as are done that are mentioned in their proprietary methodology. 3 drops were also mixed into an RNA later, which was frozen at minus 80 degrees centigrade for further use, which was considered the gold standard. So here is a diagram that is showing you the methodology of how the various samples were used. There were 13 malignant effusions. The top layer, the top part of the diagram does show the different fixatives that were used. The RNA later for fresh frozen effusion samples, cell block which were fixed in formalin, and then cytospin slides that were fixed in Cornoy, 95% ethanol and Diffquick, and then liquid-based preparations like thin prep and sure pack. The next row shows the kind of RNA extraction kit that was used for each of these samples. For the fresh frozen effusion, Coagen Mini was used. For cell box, it was Norgine. For the cytospin slides, both Norgine and PicoPure were used. However, for the purposes of analysis, the transcripts that were obtained from PicoPure were used. Then the RNA that was extracted from each of these samples underwent targeted RNA sequencing for 47 genes that were mentioned before. Here it is again, the methodology for RNA extraction. For fresh frozen sample, it was RNA-easy. For paraffin-embedded formalin-fixed tissue, it was Norgine RNA purification kit. And for the cytospin and liquid-based preparation, it was both PicoPure and Norgine. However, PicoPure RNA extraction kit was what was used for final analysis. After extraction of the RNA was concentrated, it was quantified. Its quality was then analyzed. To analyze the quality of the RNA, the authors basically calculated the number of base pairs that were present in each of these in each of the RNA samples that were obtained. So they calculated the proportion of RNA molecules that were longer than 1,000 bases, DV1000, more than 500 bases, which was DV500, or more than 200 bases. After extraction, after quantification, and analyzing the quality of the RNA sample then underwent targeted RNA sequencing with the molecular identifiers that are mentioned here, and then the resultant libraries were quantified with a bioanalyzer. For statistical analysis, the authors used Lind's concordance correlation coefficient. And I'll ask you another question here, Dr. Sura. Can you go over some of the statistical methodology that was used here, especially the CCC and the row and bias for our audience?
Sure. The Lens concordance correlation coefficient is CCC. That was used to assess the agreement for individual gene expression and breast cancer signatures of the different preparation methods to the fresh frozen sample, because we consider the fresh frozen sample to be like the ideal sample where we would get the highest RNA integrity and the best sequencing results. So what this means is that we assume the highest quality samples were going to come from the fresh frozen specimen stored in RNA preservative, and then compare that to the performance of each of the other preparation types to the fresh frozen preparation to see how that performance would stack up against it. If the correlation value is close to 1 or the CCC value is close to 1, that means that it performs similarly to the fresh frozen sample. And then the farther away from 1 that you get, that's more discordant you are from the fresh frozen sample, the most ideal sample. Some of the other statistical methods that we used were correlation or row, and the gene's deviation from perfect agreement with the fresh frozen sample, or bias, and that was used to explain lower CCC observations.
Thank you. So these are the results. I'll hand over to you, Dr. Sura, to go over these interesting results.
So this table demonstrates the results from our comparison of 2 different RNA purification methods, the Norgine and the PicoPure kits, which are both based on SIM column chromatography. The quality of the RNA was measured by the fraction of RNA fragments measuring 200 bases or more, which we designated as DV200. And we noted that the fraction of RNA fragments measuring 200 bases or more were 24% higher from cytospins fixed in carnoid solution, or 95% ethanol, than the DV200 from the fresh frozen paraffin-embedded cell blocks. SurePath samples failed our RNA-seq quality control, so we excluded that from further testing down the line. And in conclusion, we determined that the PicoPure kit outperformed the Norgine kit for extraction of RNA from the cytology preparations. Can go to the next slide. Then we compared the RNA quality for each preparation condition compared to the quality of RNA extracted from fresh frozen tissue or fresh frozen samples. The samples preserved in carnoid solution and 95% ethanol had comparable quality RNA extractions, but the FFPE and other cytology preparations had lower quality extractions compared to the fresh frozen sample. You can go to the next one. Then this slide demonstrates the concordance analysis of the 2 breast cancer signatures that Dr. Walia went over earlier, the set ERPR and set 4, or PIK3CA. There was high concordance of gene expression measurements with fresh frozen samples using cytospins fixed in carnoid solution, 95% ethanol followed by Disquick and spin prep. Then concordance was lowest for the FFPE cell block. The stingray plots on the right are to help visualize the distribution of concordance for each preparation method to the fresh frozen sample. The tighter the distribution of concordance, the smaller and closer to the top that white bar will be to the top of the line. And just the tighter and shorter the stingray plot will appear. And next slide, please. Then we have these two graphs that demonstrate the visualization of the concordance analysis of the two breast cancer signature scores, PIK3CA and SET-ERPR index. On the top graph, labeled A, are scatter plots comparing scores between different sample preparation methods and the fresh frozen samples. The closer each point lies along that line, then the more concordant the sample is to fresh frozen RNA. So you can see that the cytology slide preparation plotted more tightly to the line than the FFPE sample, which is seen with the red dots. You can appreciate that the dots are more scattered along that graph. And then in the lower graph labeled B is a visualization of the two coefficients of concordance correlation value, rho and bias. The correlation coefficient, or rho, is along the y-axis, and the bias coefficient is along the x-axis. And you can see that the FFPE samples, which are represented by the red dot, had a lower correlation and bias because the gene expression deviated more from perfect agreement with the fresh frozen sample than the other cytology samples, carnoids, ethanol, Disquick, and spin prep. Then this final figure is a figure that is, it's a comparison of transcribed mutant allele fractions between various cytologic preparations, the FFPE, carnoids, Disquick, ethanol, and spin prep, compared to fresh frozen samples. And we saw that allele fractions of most mutations are comparable between fresh frozen samples and all other methods. As you can see that the dots representing each method are hugging that trend line somewhat tightly. That's it.
Thank you.
And this is just a like quick summary that you could use to go back on, but I just went over it.
Thank you. So to summarize, the fraction of RNA fragments that measured 200 bases or more were 24% higher in cytospins that were fixed in carnoid solution, or those that were fixed in 95% ethanol, as compared to formalin-fixed paraffin-embedded cell blocks. It was also noticed that SurePath samples failed RNA sequence quality control, and hence they were not further assessed after RNA extraction. There was a high concordance of gene expression measurement with fresh frozen samples using cytospin fixed live in all the preparations, but it was lower for formalin fixed paraffin embedded cell block. And the number or the proportion of mutant transcripts was concordant with all cytological preparations. So Dr. Sura, I did want to ask you that, do you think there was different extraction kits used for different preparations? Did that affect the results in any way?
So we had compared RNA purification kits in a previous paper by Michał, Marczyk, and all in BMC cancer. The PicoPure was not compatible with the fresh FFPE sections, but it was a better alternative to Norgine for small cytospin slides. We also used the PicoPure on the direct smear FNAs in another paper with that same first author. I think you would have to do more like a direct comparison to see, but from what we saw, I don't think that the FFPE lower quality had to do necessarily with the different purification method. I think it was because it was lower quality RNA that was being extracted from the FFPE.
And in the paper, it also mentions that thin preparations had restricted range of measurement. What does that exactly mean?
The thin prep slides did not perform as well on the SET ERPR assay or the PIK3CA assay in terms of RNA expression values, and they actually had the lowest average performance on targeted RNA sequencing. They were noted to be less cellular than the other slide preparations, which we think is the reason why it led to less RNA yield. We think that the low cellularity may have been a result of suboptimal cell adhesion to the glass slide after preservation in Cytology, or it may have been a result of the vacuum transfer filtration method. The RNA extraction from that preparation had a smaller proportion of long molecules, or DV1000, compared to the high concentration alcohol-based cytospin preparations, even though they were both Pap and Nicolau stains, so we don't think that it was the Pap stain.
Thank you. Okay. So very interesting results. Cytology effusion specimens are a good source of high-quality material for nucleic acid extraction and RNA sequencing analysis. So everybody does use FFPE cell blocks because they're the go-to for everybody. But however, the study shows that the cytospin slide preparations represent a viable and probably a better option for molecular testing. And like it was mentioned that the pure RNA isolation method might be better for the cytospin preparation as compared to the Norgine method. So this study concluded that cytospin slides that are fixed in high concentrated ethanol-based fixators like cornoids and 95% ethanol, they yield superior quality RNA compared to methanol-based fixators like ThinPrep, Cytolite, or DiffQuick, or fixators that have a mixture of formalin in them, like SurePath or Cytorich, or any formalin that is present as in formalin fixed tissue as well. So these findings demonstrate that ethanol may be a more optimal for preserving biomolecular integrity than methanol or formalin. Like Dr. Sura mentioned, ThinPrep slides were less cellular than other cytospin preparations that did lead to a lower RNA yield. The authors postulated this may be a result of suboptimal cell adhesion to the glass sides following preservation in Cytolite, or maybe as a result of vacuum transfer filtration methods. The other liquid-based preparation, that is SurePath, that was used did not perform at all well in this study, and it is probable that the RNA quality in these samples was compromised due to presence of formalin in the SurePath fixation solution. So other pre-analytic considerations that the pathologists may have when they're using cytology specimens for molecular testing is the kind of slide that is being used. So non-frosted and positively charged slides in prior studies have shown to yield higher amounts of DNA, and also cover slip removal techniques with xylene or liquid nitrogen prior to extraction of DNA and RNA have been shown not to affect the quantity or quality of the molecule. The authors chose materials from scraped cytology slides and not material from supernatant because it was just to be consistent with a microscopic review of stained slide for selection. Like Dr. Sura mentioned, most of them did have more than 10% cellularity for tumor cells. However, it is an option to use the stock fluid that is stored. It just has to be spun down and concentrated to a more usable form before sending it for molecular testing. So obviously, this was a study that was done under supervised research protocol. It does not represent how labs usually get their samples and handle their samples. So that is one limitation of the study. And another limitation of the study is that it was not longer storage times, or the effect of longer storage times was not taken into consideration. So if any of the cytospin slides have been stored for a longer time, does the nucleic acid degrade over time? And that was not studied in this and needs to be further looked into. So in conclusion, the success of molecular testing on cytology samples is highly dependent on pre-analytical factors. Cytospin preparation slides with cornoids are 95% ethanol and then stained with Papa-Nicolau stain produce RNA sequencing results that were equivalent to fresh frozen samples and superior to hormone-fixed paraffin-embedded cell rock sections. That's all I have. Thank you. Any questions?
Thank you very much. This was very nice and very well organized to kind of tell us all the ins and outs of how the article was made to come to life, and also how the clinical validity of using the cytology specimens. I do not see any questions in the question box. So that's usually a good thing, I think, because that means that whatever you guys said, it was very well understood. That's perfectly okay. I just wanted to kind of say again what you guys were saying. Cytology specimens are really a great source for doing everything from the day that cytology was introduced as in pathology, because it's amazing how much we can get from just looking at individual cells in the background of so much blood and so much other material. So that's the reason that I was very much interested in cytology after my first rotation, which during frozen sections, we were using a lot of cytology touch preps and smearing of the lesions. It was a different timing. I'm not going to say how old I am because that would give it away, but it was a time that there was not much of the EBUS or EUS or, all these pre-surgical interventions that nowadays are done. So a lot of patients, when you have gone to the surgery, because that was the only way to get some sample. And the samples, we had to make diagnosis on the spot. And that was kind of scary, but at the same time rewarding, because I got to see cytology, how powerful it is. And so I'm very happy that over the years, it's been more and more become, a popular methodology, if I should say, for looking at things in a very non-invasive manner. And we now do all sorts of the immunohistochemical stains on the cytology material. We do all sorts of the molecular studies on the cytologic material. So it is great to see that the trend is continuous upward, and that I think that we see it every day. I mean, when we get all of these EBUS samples, many times the transbronchial biopsy that comes with a lung, for example, mass, or a lymph node, mediastinal lymph node, we had better material in the cytology. So that's what's used for the biomarkers and for the immunos and everything else. So I think that's a really strong statement to the power of cytopathology for the diagnostic and for prognostic and everything after for the patient care. So as I said, I don't see any questions at the moment. We usually tell the audience, if you have burning questions that you didn't get to ask, they can send an email to, I think your email, Dr. Walia, was on the paper, on the title, or no?
No, it wasn't. But
okay.
To Ms. Jenkins, or maybe they can forward it to me.
Yeah, I think they can figure out if they need to reach out, and so I think the questions can be answered that way as well. Any last minute thoughts or statements that any one of you wants to make?
I just wanted to thank Dr. Zai and Dr. Sura for being here and discussing this article. I think it is a very important article. Like I said, I did the poll yesterday, and most people were considering just tumor cellularity as a, to figure out which specimen to send from molecular testing, and also almost everybody was using formalin fixed tissue. So I think the study is very important to say that we have to look at other pre-analytical factors as well to make sure we can get optimal test results, especially if they're being used for, targeted treatments for patients. So very important results, I think, for the cytopathology community.
Yeah, and thank you for choosing our article and going through it so diligently. I know it's a lot of material to digest, but I think it has a great take-home point that we can use cytospin slides fixed in high concentration alcohol. You don't have to rely heavily on FSCE. There's so many situations where there is nothing in the cell block, even when you've done adequacy and you have all the tumor on the cells. So if laboratories are able to validate the use of smears for molecular testing, it could save a lot of patients. An extra procedure, it could save a lot of time to diagnosis. It's a great thing, and it's not necessarily hard to do. So yeah, thank you all for your time, and had a nice time joining you.
Very good. Really appreciate everybody and the audience who are listening, and also the two of you who are present in flesh here. So I think in the interest of time, we can kind of say goodbye at this point. And thanks again for your hard work, both for making the presentation of the article and also for putting out your finding in the article that it can be looked at and referenced to for future to come. Very good. Have a wonderful day, and hopefully to see many of you or some of you in Austin next the next week or so. Right. Very good. Have a wonderful day. Bye-bye. Bye.